Whenever you see an fMRI image of the brain in a journal article or a movie, you probably only see one or two blobs of activation. These blobs are thresholded, meaning that the activation is strong enough to be statistically significant. In other words, we wouldn't expect to see these blobs just due to chance. However, this leaves out all of the other effects in the brain that did not pass the threshold. In effect, they are hidden. But does that mean there isn't anything there?
You have no activation in your amygdala.
A recent article by Paul Taylor and colleagues in Neuroimage suggests that there's a better approach, highlighting the data, first proposed by Elena Allen et al. In 2012. This means that we clearly indicate which blobs or voxels pass our threshold, while also showing the rest of the data using translucent maps. Those voxels that have stronger effect sizes are colored brighter, while weaker effects are more transparent. To illustrate this approach, the authors used the data from a recent study by Botvinik-Nezer and colleagues published in Nature in 2020. In this study, 70 independent teams of researchers analyzed the same data set, a neuroimaging study of gambles. Because most studies like this find activation in reward-sensitive parts of the brain, including the ventral striatum and the ventral medial prefrontal cortex, each team was asked to analyze the data however they wanted, and then report whether they found an effect in any of those regions. Instead of a consensus like we would expect from multiple teams analyzing the same data, the amount of agreement between the researchers was actually quite low. Consider, for example, this figure of the thresholded maps for each group and the similarity matrices showing the overlap between each group's statistical maps. At first glance, there appears to be quite a lot of variability in the results. However, if we use the highlighting approach, we see much more agreement between the groups. This suggests that virtually all of the researchers are finding the same effect, it just doesn't pass everyone's threshold. So why is this highlighting approach important? First of all, consider the issue of reproducibility in the sciences, or the ability of independent researchers to obtain similar results. Using the binary significant or non-significant approach, researchers are constrained to either say that the result is there or it isn't. But it could be that while Team A, for example, finds a significant effect in one part of the brain, Team B has a similar trend in the exact same area. But maybe their study is underpowered from scanning fewer subjects than Team A. In this scenario, Team B may mistakenly conclude that they were unable to replicate Team A's finding, and general confusion follows. This can obscure the full picture and mislead us, while highlighting, on the other hand, allows the reader to make a more educated decision about the evidence for or against an effect. Here is a short demonstration of how to highlight your results in AFNI. First, overlay your statistical map on a template, select your threshold, and then click the A and B buttons above the threshold slider. This will make the results translucent and also highlight the significant results with a black border. You can then make further edits like resampling to make the clusters look smoother. Just include it as a figure in your paper and you're done. So there you have it, an overview of the highlighting approach for displaying neuroimaging results. And now I want to hear from you all about what you think about this paper and their approach. Do you think it's a better way of presenting the data than the traditional ways of doing it? Do you think it should be required for publication? Are there any disadvantages of this approach that we haven't thought about yet? And do you think it's going to have any kind of noticeable impact on reproducibility if everybody starts doing this? Do you think it might discourage practices like p-hacking, where you keep modifying your pipeline until you just get a result that just passes that significance threshold? Let me know what you think in the comments down below.